Hi everyone, my name is Michael Amoka and I'm going into my third year majoring in biomedical engineering. Currently, I am on a co-op in Dr. Wirch's lab under Petra's project. Now, this project focuses on understanding the underlying molecular mechanisms of toll-like receptors, in our case specifically TLR2 in degenerating intervertebral discs. Here is a quick overview of what I am going to talk about. So starting with the background, the first thing I'm going to be talking about is intervertebral discs. Now, these are located in between the vertebral body and can act as a cushion to absorb shocks and stresses from our daily movements. Now, with these, there are actually three main components. However, we are only interested in the annulus fibrosus and the nucleus pulposus. As you can see on the right, the AF is the outer layer. This layer is more rigid and can help with rotational stability and resisting compressive forces. The NP is the inner layer, which is a more gel-like substance, which can help transmit stress and force from vertebrae to vertebrae. Now, moving on to degenerative disc disease. This is very relevant and important since it is one of the main causes of low back pain. And with low back pain, this can actually really negatively affect someone's quality of life to the point where they may not be able to provide for themselves and ultimately take care of themselves. Even though it's very important, the existing treatments today are actually based on just relieving symptoms. One reason is because we don't fully understand the underlying mechanisms yet. So another thing to note is with deterioration, this actually happens on a tissue, cellular, and molecular level. One example is with extracellular matrix degradation, which can lead to inflammation and pain. However, based on the degree of degradation, this inflammation and pain can vary from person to person. As you can see in figure 4, there are 4 different degrees. So at the very top, that is minimal degradation from a relatively healthy disc. And as you go down, the degradation gets worse. Moving on to toll-like receptors, we're specifically going to focus on TLR2 since that is what's prevalent in IVDs. Now, these are intermembrane proteins that regulate the inflammatory pathways. An example of this pathway is shown on the figure on the right. They could also be activated in 2 different ways. The first way is the stronger way to activate them, which is the pathogen-associated molecular patterns. The second way is the damage-associated molecular patterns. Lastly, we have microRNAs. Now, these microRNAs are very short regions of non-coding RNA. However, they can still affect gene expression. As you can see in figure 4, these mRNAs can bind to something called a RNA-induced silencing complex, otherwise known as a RISC. And then this can bind to the mRNA and affect a specific gene expression. Now, depending on how well they bind to the mRNA, there are different effects. So if it's only a partial match, the expression is repressed until the mRNA is degraded. However, if it's a more extensive match, the mRNA is rapidly degraded. These are also reusable and can bind to multiple genes, making them a very strong regulator of gene expression. Now, before we move on, I wanted to mention a research objective for this presentation, which has to do with validating TLR2 associated microRNAs, validating TLR2 knockdown, and also validating its activation. To do this, we activate TLR2, we harvest and isolate the mRNA, perform a RT-qPCR, and then use an ELISA to confirm the activation of TLR2. For the previous work, TLR2 knockdown with small interfering RNA was established. These work similarly to mRNAs as they also bind to a RISC protein. However, instead of only being a partial or extensive match, it is a perfect match, which leads to mRNA degradation. We also activate TLR2 with PAM2 as our PAMF and fragmented fibronectin as our DAM. Another thing they did was use next generation sequencing, which was used to identify a set of microRNAs in different conditions, including PAM2 treatments and TLR2 knockdown. Now moving on to some of the methods, the first thing has to do with TLR2 activation. We start with adding our different sRNA conditions, which includes the not treated, not targeting, and the TLR2 sRNA. Then we activate TLR2 with PAM2 and fragmented fibronectin, and we also have an extra plate that acts as a control that isn't treated. Next, after incubation, we harvest the supernatant and use that for the ELISA. And then we add chiasol lysis reagent, which we got from QIAGEN, to lyse the cells open. These cells are detached using a cell scraper and then transferred to Epinorf2. This cell lysate is then used for mRNA acylation. For this, we use mRNA Easy Kit from QIAGEN. And the first step involves a liquid to liquid extraction where we will see a phase separation. From the bottom, there is the organic phase. Then there is a small white interphase, and then the top is the aqueous phase, and that is the phase we are interested in. This is then transferred to a column and then washed until the mRNA is eluted. We can then use a nanodrop to quantify the yield and purity, and this purified mRNA is then used for the reverse transcriptase step of our RT-qPCR, and this is used to synthesize our cDNA, which is the complementary DNA. For this, we use a RT kit from QIAGEN, and at the bottom left, you can see the reagents needed for this reaction. This includes the mRNA we isolated, which is the template RNA we are going to use. The figure on the right shows the basic reaction. To quickly go over it, there's the poly-A polymerase, which adds a poly-A tail to the mRNA. This allows the poly-T primer to bind to the poly-A tail, which allows the reverse transcriptase to bind and synthesize the cDNA. This cDNA is then used for our next step, which is the qPCR amplification. And for this step, we use a cyber-green PCR kit from QIAGEN. And like the last slide, the reagents are shown in the bottom left. One important thing to note about this, though, is the PCR primer does depend on the microRNA of interest. This is because the primer itself is specific to the microRNA. It will amplify the specific microRNA, and then that's where we will get our reading. After pipetting this reaction into a PCR plate, we then put it into a qPCR machine. And on the right, we could see a brief overview of this reaction. So the first step has to do with activating the DNA polymerase and then denaturing the DNA. The second step has to do with annealing the primer and then extending it. During this, fluorescence is measured, and then this cycle is repeated 40 times. For the final step, this is the final step, the data we want is the CT value. The CT value is the number of cycles it takes to reach a certain level of gene expression, which is measured through the cyber-green fluorescence. We can then calculate the relative fold change from here. Now, to validate TLR2 activation, we use a sandwich ELISA protocol with either interleukin 6 or 8 from R&D. We use these two because IL-6 and 8 are commonly secreted in the inflammatory pathway when TLR2 is activated. So the first step is to coat the plate with a capture antibody, which is specific to IL-6 or IL-8. And then we add a blocking buffer to make sure that nothing else can bind to the surface. We then add the samples and standards, which contain the interleukins that can bind to the capture antibody. The detection antibody can then bind to the interleukin, which allows strep HRP to bind. So when we add our TMB substrate, a colorimetric reaction occurs as it interacts with the strep HRP. Next, we add a stop solution, which stops this colorimetric reaction. And finally, we could bring this plate and measure its absorbance and compare it to the absorbance of the samples to interpolate the unknown concentration based on our standard curve. Now, moving on to the results. As of now, the results are going to be focused on validating TLR2 knockdown and activation, since we are still analyzing the gene expression data. So for the knockdown validation, on the figure on the left, we can see a significant difference in the relative fold change. Compared to the non-targeting TLR2 gene expression for the TLR2 knockdown is significantly reduced. On the right is the same results, however, it's quantified by percentage, and we can see that there is almost a 78% TLR2 knockdown. This validates that our knockdown is working. Now, to validate TLR2 expression, we look at the IL-6 and IL-8 from our ELISA data. Since these are commonly secreted with TLR2, we use IL-6 and IL-8. So if there's a significant increase compared to the control, we can say that TLR2 was activated. For these results, both IL-8 and IL-6, for both non-targeting and TLR2 knockdown, we can see an increase in IL-6 and IL-8 secretion, which implies that TLR2 is activated. And one last thing to note is as of now, we don't have the data for the fragmented fibronectin, so this is just the data for PAM2. Lastly, we have our future work. So with that, of course, we have to complete the gene expression analysis. In addition to that, we have also started optimization of TLR2 knockdown using CRISPR-Cas9. Another thing we want to do is check on the functionality of AF cells in a 2D culture, and also check the microRNA functionality in NP cells in a 3D hydrogel culture. And the last thing would be, since these cells are mechanosensitive, we want to see if placing these cells under mechanical loads will actually change the gene expression or mRNA functionality. Now, here is a list of my citations. And thank you guys for listening. If you have any questions, feel free to reach out. My email is listed right below, or you could also comment on this video. Thank you.